Today we are going to discussing about mutations. We are discussing about two types of mutations. First one is spontaneous mutations and second one is induced mutations. First we are going to discussing about spontaneous mutations. Spontaneous means sudden change, sudden mutations, sudden changes. Mutations are heritable changes in the DNA. They are essential to the study of genetics and are useful in many other biological fields. Somatic mutations means this type of somatic mutations occurs in non-reproductive cells. Non-reproductive cells means asexual reproductive cells. Asexual reproductive cells. And second one is germline mutations. Germline mutations means this mutations occurs in only cells that give rise to gametes. The germline mutations is also called as sexual and reproductive cells. This type of mutations occurs in reproductive cells. Some spontaneous mutations caused mainly by spontaneous replication errors. All these different types of spontaneous mutations. First one is cytomeric shifts, and second one is bell base pairing, and third one is strand slippage, and fourth one is unequal crossing over, and fifth one is spontaneous chemical changes. Spontaneous chemical changes are two types. First one is depurination and second one is deamination. Spontaneous chemical changes, depurination, the loss of a purine base from a nucleotide. This is called as spontaneous. This is called as depurination. This is the one type of spontaneous chemical changes. And now we are going to discussing about induced mutations. Mutations are those that result from changes caused by environmental chemicals or radiations are called as induced mutations. A number of environmental agents are capable of damaging DNA, including certain chemicals and radiations. ఇక్కడ మనకి ఏంటంటే డిఎన్ఏ అనేది డిఎన్ఏలో వచ్చి చేంజెస్, సడన్ చేంజెస్ లేకపోతే వాటిలో ఉండే యొక్క కెమికల్ చేంజెస్ అనేవి మనం ఎక్కడ అబ్జర్వ్ చేస్తామంటే మనకి ఇక్కడ మెయిన్గా certain chemicals and radiation, అంటే ఫిజికల్ అండ్ కెమికల్ టూ టైప్స్ ఆఫ్ చేంజెస్ అకర్స్ ఇన్ DNA. The type of mutations is called as induced mutations. Mutation, an environmental agent that significantly increases the rate of mutation above the spontaneous rate. And next, the first discovery of chemical mutation was made by Cholret and Auerbach. Induced mutations are different types caused by base analogs, alkylating agents, deamination, hydroxylamine, oxidative reactions, intercalating agents, radiation. All these are induced mutations. First we are going to discussing about base analogs. Chemicals with structure similar to that of any of the 4 strand bases of DNA. DNA polymerases cannot distinguish these analogs from the standard bases. So, if base analogs are present during replication, they may be incorporated into newly synthesized DNA molecules. Examples: F-bromo, uracil. And next, alkylating agents. Chemicals that donate alkyl groups. These agents include methyl and ethyl groups, which are added to nucleotide bases by some chemicals. Example: ethyl methane sulfonate adds an ethyl group to guanine, producing 6 ethyl guanine, which pairs with thiamine. Deamination. Deamination means the loss of amino group from a base. Deamination may occur spontaneously or be induced by mutagenic chemicals. This is also one type of spontaneous mutations. Hydroxylamine. Hydroxylamine is a very specific base modifying mutation that adds a hydroxyl group to cytosine. It converts cytosine into hydroxylaminocytosine. This conversion increases the frequency of rare tautomer. The tautomer pairs with adenine instead of guanine and leads to CG-TA transitions. Because hydroxylamine acts only as cytosine, it will not generate thiamine and adenine and cytosine and guanine transitions. Radiations. Ionizing radiation breaks covalent bonds including those in DNA and is the leading cause of chromosomes mutations. Ionizing radiations also frequently results in double strand breaks in DNA. Ionizing radiation has a cumulative effect and kills cells at high doses. UV causes purines and pyrimidines to form abnormal dimer bonds and bulks in DNA strands. Examples UV and X-rays, etc. Oxidative reactions. Reactive forms of oxygen damage DNA and induce mutations by bringing about chemical changes to DNA. Reactive forms of oxygen include superoxide radicals and hydrogen peroxide and hydroxyl radicals. They are produced in the course of normal aerobic metabolism as well as by radiation, ozone peroxides, and certain drugs. And next, intercalating agents. Intercalating agents produce mutations by sandwiching themselves between adjacent bases in DNA. They distort the three-dimensional structures of the helix and causing single nucleotide insertions and deletions and replication. These insertions and deletions frequently produce frameshift mutations, and so the mutagenic effects of intercalating agents are often severe. Because intercalating agents generate both additions and deletions, they can reverse the effect of their own mutations. Examples proflavin and ecridine, orange and ethidium bromide and dioxin. These are examples of intercalating agents. Thank you.